Welcome to my crib.
Oh.
I'm just grabbing the DNA. So I did a gDNA extraction yesterday for genomic DNA.
Oh.
Wow. So I did it on 5 animals of these tiny aplacophorans species, Virenia argentea. And we need about 200 nanograms for PacBio sequencing. So this is the wrong team. We're hoping not to pull individuals together, so we're trying to see if we have enough. And preliminarily, it looks like I do, but now we have to visualize the DNA to see if it's degraded or not. And so we'll get a gel image, something hopefully like better than this. This is the last time I did Virenia, not like this, because this is really streaked. It seems like I do have enough DNA right now, but we just got to make sure that it's all like good DNA.
So each one of these tubes is an individual?
Yep. 1 through 5 are Virenia argentea. They're really small, so we did a lot just to have like replicates to see. These are starved individuals, which is awesome because it will help with contamination issues because they eat cnidarians, and you can have like cnidarian contamination.
Yeah, so I think a lot of people don't realize that yeah, when you
sequence a whole thing, because they're so small, you have to sequence the whole animal, and
right.
So you're also sequencing whatever it ate. You get bacterial contamination.
You get bacteria, you get yeah. And usually you can
bioinformatically take that out. And then the other one is another Caudophobia apacophoran, and that is Scutopus ventrilineatus from Norway. So this is a gel.
Okay, so let's check this out.
Okay, so your gel is agarose, basically.
Yeah, it's like a jello, basically.
It's gel. I don't know how else to say it other than that it's gel. And this little mold right here, there's going to be little wells, and we're going to load those wells with
those
our
are like
DNA.
little teeth that stick into it.
Yep. You load a ladder in, which basically is like a known amount of kilobases of DNA. So this is a ladder, so it'll tell you wherever your DNA kind of goes out on the gel. You can be like, okay, so I know that this is 2,000 base pairs. I know that this is 100 base pairs kind of thing.
Yeah, so you can line it up.
Exactly. DNA is negative, so it is going to go towards the positive side, essentially. And it'll stop migrating at however many base pairs that it is.
Yeah, because
it'll all parse out.
Because large pieces move slower than the big pieces. Okay, so you just took the little comb out.
Yep.
So those are the holes. The little orange dot?
Yeah, so that's just the loading die, and then I'll add the ladder.
Cool, so now all those are yellow.
Yep. Then they give you a genome.
And then you set a timer, right?
Yep.
So it runs at a certain voltage.
Run, and you should see bubbles start to form.
Okay, so we're waiting on the...
We're waiting on the gel.
Yeah, we're waiting on the gel. And so that's you said
Fingers crossed, am I right?
Yeah. That's you said your genomic DNA.
Yes.
From aplacophorans?
From aplacophorans.
Okay, and so we've talked about them before, that Maddie has mentioned that they're worm-like mollusks that are...
Yes.
What's the proper term? Basal, early branching? What's the...
It's not basal.
Okay.
Early branching.
Okay.
And so your aplacophorans come in 2 different flavors. Maddie studies selenogasters.
Okay.
So selenogasters have a foot. That is kind of the main distinguishing factor. And caudophobia do not. And those, together with chitons, which like our lab mate Rebecca studies, they form this clade Aculeifera. And this clade Aculeifera is sister to all other mollusks, which are conchiferans. So apacophoran means without shell, right? Polyplacophoran, which are the chitons, means many shells.
So is this related to, like...
This.
A scallop shell or something?
Exactly. Aculeiferans, so the chitons, and then our like favorite little guys, aplacophorans, are covered in these like spicules. They're called sclerites. And so what my question is, are these little, because it's calcium carbonate, it's like aragonite, are these calcium carbonate sclerites, is the genetic toolkit the same as it is to make these little sclerites as it is to make a shell in like a snail or something. And so how we're doing that is we're sequencing genomes.
Yeah.
And for example, nacre is what makes like abalone shiny towards the bottom, or like pearl oysters and stuff like that, shiny on the inside.
Yeah.
Nacre has evolved like 5 different times over like bivalves and gastropods. So like just because you're making this mineralized structure and it's like made out of the same stuff doesn't mean that the way that you got there is the same.
Yeah.
So they could be independently derived.
Right.
So the ones that are on the gel are these Virenia, and then I did 5 of them, and then a Scutopus, and then my last one that I'm doing is Chaetoderma natigulum, which is a relatively large caudifoviate.
Gotcha.
Yep.
So, you want to go check the gel?
Okay. So that's the dye that we're in.
Oh yeah, so that little band in the middle.
Yeah.
So this is UV, so the orange part is to not kill ourselves.
Oh wow, that is straight up disappointing right there.
So on the left
It's the ladder.
is the ladder.
So I
And
got a lot of DNA, just did not get a lot of good DNA. So there's faint bands, so like we definitely have DNA, but it's just not the best. We're not happy. Just yeet. Sad yeet. Wasn't it really just about the friends that we made along the way?